Hello students! Today you've started learning medical microbiology, and we start from general microbiology. Today we'll discuss history of microbiology, classification of bacteria, structure of bacterial cells. Why study microbiology? You can see several points like pharmaceuticals, bioremediation, and pathogenicity. Medical microbiology is the basis for a process of vaccination. It's protection of society against the infectional diseases, severe, dangerous diseases. Medical microbiology is understanding all principles of chemotherapy and antibiotic therapy. Biotechnology can be used in our ordinary life for our protection, for improving our health. Bioremediation is a possibility to protect environment or to improve environment. And pathogenicity may be one of the most important things. If you understand pathogenicity, you can understand how you can treat your patient, how you can cure your patient, because you can make correct diagnosis. Microorganisms is living things too small to be seen by naked eye, and the name of microorganisms is similar to microbes. You can see that all microorganisms divided into 6 groups: bacteria, archaea, algae, fungi, protozoa, and viruses. There are 4 of them are medically important. It's bacteria, it's fungi, protozoa, and viruses. All these groups of bacteria can cause infectional diseases. History of microbiology. Microbes discovered more than 300 years and known to man during the middle of the 18th centuries. Period of progress began and continues to nowadays. And periods of microbiology development: morphological, physiological, and prophylactic periods. Morphological period of microbiology history was started in middle of 17th century. It's also called micrographical period, as the study of microorganisms came only to description of their dimensions and forms. Biological properties and their significance for man still a long time remained incomprehensible. Experimental phase connected with names of scientists like Leeuwenhoek, Pasteur, Koch, and Lister. Before 17th century, study of microbiology was hampered by the lack of appropriate tools to observe microbes. Robert Hooke in 1665 built a compound light microscope and used it to observe thin slice of cork and coined the word cells. Anton van Leeuwenhoek in 1673 was the first person to observe live microorganisms, which he called animalcules, bacteria and protozoa, using single-lens microscope that he designed. He observed 50,000 different specimen reported findings to the Royal Society of London. On this slide you can see the portrait of Anthony van Leeuwenhoek discovers microorganisms. Edward Jenner is the father of vaccination. He was first scientist who created vaccine, and it was vaccine against smallpox. And his portrait. Pasteur's contribution. Process of pasteurization developed a process in which liquids are heated to kill most bacteria responsible for spoilage. Disease causes. Identified 3 different microbes that caused silkworm disease. He created vaccine, developed a vaccine for rabies from dried spinal cord of infected rabbits, and directed Pasteur Institute until his death in 1895. And the portrait of Louis Pasteur that in 1859 disapproved the spontaneous generation of microorganisms. And exactly the experiment. Joseph Lister, 1959, used disinfectant to treat surgical wound, greatly reducing infection rates, and considered the father of antiseptic surgery. And his portrait. Robert Koch. This scientist named like a father of medical microbiology. First person proved that microorganisms caused diseases. Only specific microorganisms caused specific diseases. He studied anthrax, affects cattle and humans, proved that Bacillus anthracis cause anthrax in cattle. Later, Robert Koch identified bacterium that causes tuberculosis. Robert Koch. And from 1892 was started a rising of virology. Ivanovsky, studying disease of tobacco plants, and discovered small infectional microorganisms named like a virus. And prophylactic period connected with the name of Ilya Ilyich Mechnikov. Classic Mechnikov's research defined a prophylactic period in microbiology history. And now you see 3 surnames of scientists who discovered sulfonylamides and antibiotics. Paul Ehrlich discovered salvarsan. Alexander Fleming discovered first antibiotics, penicillin, and René Dubois discovered 2 antibiotics, gramidin and tiracidin. Alexander Fleming, with his creation antibiotic penicillin, and you see that in previous century, 1970, was started discovering of genome of bacteria. Classification system. in microbiology usually use Berger's manual. Berger's manual of determinative bacteriology actually is the Bible of bacterial taxonomy. There are such levels of microorganisms organization. You can see species, genus, family, class, division, and kingdom. 35 of the major groups of bacteria are distinguished primarily on morphological characteristics, namely cells shape, its rod, cocci, curved or filament forming, spore production, staining reaction, and modality. Other groups are defined based on their metabolism or combination of morphological and physiological characteristics. And on this slide you can see some of the major groups of bacteria in Berger's manual. The group, for example, the group gram-positive cocci, it's usually bacteria that have a cell wall structure that result on their staining blue-purple by the gram staining procedure, and that are spherical, include the streptococci and staphylococci. What does it mean bacteria? It's small, single-celled organisms, prokaryotes before nucleus, lack of following structures: nuclear membrane around DNA, membrane-bound organelles like mitochondria, chloroplasts, Golgi apparatus, endoplasmatic reticulum, and lysosomes. On this slide you can see the table where compared prokaryotic and eukaryotic microorganisms. Different size. You can see that cell wall present in both cases except mycoplasma in case with prokaryotic bacteria, cytoplasmatic membrane present in both cases but contain sterols in eukaryotic microorganisms and absent in prokaryotic. Only one exception is mycoplasma. Nuclear membrane absent in prokaryotic microorganisms and present in eukaryotic. Nucleus absent in prokaryotic, present eukaryotic. You can see that chromosome, single chromosome in case with prokaryotic bacteria and more than one in eukaryotic. Mitochondria absent in prokaryotic, present eukaryotic bacteria. Different types sedimentation of ribosomes, and reproduction in case with prokaryotic microorganism. It's only asexual type of reproduction, and we are speaking about binary fusion, and two types of reproduction in eukaryotic microorganisms, sexual and asexual, and examples in case with prokaryotic microorganism, absolutely different microorganisms, bacteria, chlamydia, rickettsia, and eukaryotic also variety like fungi or protozoa. Bacterial identification and classification. Bacteria can be classified accordingly to the shape. It can be cocci, bacilli, spiral shaped bacteria. Arrangement: single, in pairs, in chains, in clusters. Accordingly, the size can be small, middle size, or big microorganisms. Accordingly to the gram staining, can be gram positive or gram negative types of microorganisms. Accordingly, the type of respiration, aerobic or anaerobic, can be classified accordingly to physical or structural characteristics and biochemical characteristics, and of course accordingly to DNA analysis. And on this slide you can see the dimension of bacteria. The smallest microorganisms on this slide you can see it's viruses, rhinoviruses, bigger Ebola virus, but the same time you can see that viruses much smaller than bacteria, and bacteria at the same time much smaller than humans cells. Cross morphology of bacteria. On this slide you can see that the size of bacteria also can be different from 1 to 10 micrometers. You can see again different shape. Coccus is round, bacilli is rod-shaped bacteria and can be curved bacteria, can be corkscrew bacteria. We name corkscrew bacteria like Spirillum, and flexible corkscrew bacteria it's Spirochetes. Just a little bit curved bacteria it's Vibrios. And classification of bacteria accordingly to arrangement. And you can see that can be monococcus, can be diplococcus, it's in pairs, tetrad, it's 4 bacteria, sarcine, it's clusters, and staphylococci, it's irregular clusters. And on this slide you can see Streptococcus, it's bacteria like situated like a chain, and right photo is electronic microscopy, left photo it's gram staining light immersional microscopy. And clusters of cocci you can see on right photo it's staphylococci from patient's material, and left part you can see staphylococci stained by gram staining in puer culture. It means only one type of bacteria, one genus, species of bacteria. Bacillus shaped bacteria you can see E. coli and Pseudomonas aeruginosa. There are 2 gram-negative rod-shaped bacteria, 2 types rod-shaped bacteria, but you can see that different size. Escherichia coli, it's middle size bacteria, and bigger Pseudomonas aeruginosa. And bacillus-shaped bacteria like a Fusobacterium, you can see it's much bigger microorganism, rod-shaped microorganism. If bacillus or rod-shaped bacteria situated like a chain arrangement, like a chain, we can name it like Streptobacilli. And the example of Streptobacilli, Bacillus anthracis. Palisades arrangement. You can see this arrangement, and an example is Carinibacterium diphtheria. Curved bacteria. Left part is Vibrio, it's just slightly curved Vibrio, Calere bacteria, and corkscrew Campylobacter. Curved bacteria, Spirilla and Borrelia burgdorferi, is the member of order Spirochetalis, so it's flexible microorganism, corkscrew flexible microorganism. And bacterial surface structure or morphology of bacteria. You can see that bacteria has envelope, and this bacteria envelope contain cell wall and plasma membrane. On cell wall situated flagellas, this one, yeah, flagella. So there are 3 flagellas on this scheme. You can see that on cell wall can be villi, and inside of cell wall envelope situated nucleoid side and cytoplasm, and in cytoplasm you can notice ribosomes. Sometimes around of cell wall can be capsula, can be situated capsula. It can be small capsula like так, блин, что? Cell envelope. Cell envelope contain cytoplasmic membrane and cell wall. The group of bacteria that lack of cell wall named L-form. They don't have peptidoglycan layer, and cell membrane contains sterol for stability. Cytoplasmic membrane is phospholipid bilayer fluid mosaic model, embedded proteins for active transport, enzymes for energy generation, and photosynthetic pigment. And on this slide you can see the scheme of cell membrane. Function of cytoplasmic membrane: its selective permeability to different molecules, active transport aided by permease, play a role in DNA replication, cell wall biosynthesis, and mesosomes for cell division. Cell wall. There are 2 major groups of bacteria based on structure of cell wall: gram-positive and gram-negative. Gram-positive microorganisms have thick peptidoglycan layer, gram-negative thin peptidoglycan layer, and outer membrane contains lipopolysaccharides. Gram stain is crucial first step towards identification. And on this slide you can see the structure, the scheme of peptidoglycan. Peptidoglycan contain N-acetylmuramic acid and N-acetylglycosamine connected each other by peptide bridges. Cell wall. Gram-positive cell wall. It's thick peptidoglycan, it's acidic polysaccharides and teichoic acid and lipoteichoic acid. Gram-negative cell wall, it's thin peptidoglycan, a lot of lipopolysaccharides, porins, and periplasmatic space. And you can see exactly the structure of gram-positive cell envelope. So, thick peptidoglycan and no periplasmatic space inside of peptidoglycan teichoic acid. And gram-negative cell wall you can see thin peptidoglycan, periplasmatic space, and outer membrane. Outer membrane and peptidoglycan connected each other by lipoproteins. And on this slide you can compare the structure of cell wall, gram-positive and gram-negative. And you can see that gram-negative cell wall much more complicated than gram-positive cell wall. Function of cell wall: its creation of the shape because of rigidity of peptidoglycan, its protection the cytoplasmatic membrane, and cell wall is osmotically insensitive, you have to know it. And microorganisms lost on cell wall named like L-form. And the point is there are two variants of L-form. Protoplast can be created from gram-positive microorganisms, and protoplast doesn't have any pieces of cell wall, and spheroplast created from gram-negative cell wall, gram-negative microorganism, and can contain some parts of cell wall. The example of L-form microorganism is mycoplasma. This microorganism can't reverse, can't create cell wall. Capsules. Capsules important for adhesion and for protection microorganism against the immune system of human's body, of animal body, and capsule important for protection against the dehydration. And you can see examples of microorganisms with capsule. So microscopy, you can see microscopy, Klebsiella pneumonia, you can see this capsule around of microorganism, and Streptococcus pneumonia, again capsule creation microorganism. And cultural properties of microorganisms that have capsule, it's this mucoid type of colonies, and one of microorganisms can create capsule, can synthesize capsule, is Bacillus anthracis. External structures. There are several variants. It's pili or fimbria play roles in adhesion of microorganism to host cell, exchanging of genetic material, and in this case we name this pili like F. pili because of fertility and for avoidance of immune response. Flagella are important for motility, and flagella is antigenic determinant, so it's H antigen. It's important for immune function of. On this slide you can see photos of microorganisms with pili and flagella. You can see the process of attachment E. coli to host cell by pili. You can see scanning electronic microscopy of Salmonella. You can see the smaller organelles, it's pili and long flagella for motility. And F. pili, it's for organelles for exchanging of genetic material, genetic information from one microorganism to another one. Flagella structure. So you can see that flagella contain part like a basal body, and this basal body contain rings, can be contain actually the pairs of rings, can be one pair, can be two pairs of rings. It depends on type of cell wall. Again, gram-negative microorganisms have two pairs, it means 4 rings, and gram-positive microorganisms have only one pair of rings, only 2 rings. After this basal body, you can see hook and exactly filament. Flagella composed of protein called flagellin. And arrangement of flagella. You can see only one flagella, it's monotrich. Lophotrich is several flagellas on one side. Peritrichas, it's microorganism with flagellas around of whole body, and amphitrichas is microorganism with several flagellas in 2 sizes. And you can see bacterial motility also quite different. Can be motility by rotation, can be like a slate bacteria, so different types. And internal structures like cytoplasm, genome, inclusion bodies, and endosperm. Cytoplasm is gelatinous solution containing water, nutrients, proteins, and genetic material. Site for cell metabolism. And chemical analysis of microbial cytoplasm. 70% is water plus proteins. 96% of cell is composed of 6 elements: carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. And you can see that most bacteria contain a single circular double strand of DNA called a nucleoid. Prokaryotic ribosomes. A ribosome is a combination of RNA and protein and is the site of protein synthesis. There are 2 subunits, 50 and small 30 sedimentation coefficient. Inclusion bodies enable a cell to store nutrients and these organelles for surviving of microorganisms in environment. Bacterial spores. Some bacteria, and actually we are speaking about genera Bacillus and Clostridium, develop a highly resistant resting phase of endospore that does not grow or reproduce and exhibit absolute dormancy, no detectable metabolism. And you can see on this slide endospores created by Bacillus anthracis. Vegetative form. The bacteria actively growing non-spore stage of a bacterium. Sporulation is formed on exposure to unfavorable condition. It can be nutrient depletion, it can be changes of moisture, temperature, pH, or oxygen tension, and spore requires 10-15 hours to form. And on this slide you can see the scheme of endospore formation and the process opposite to sporulation, germination. Mature endospore are metabolically inert. Changes in the environment, like returning to vegetative state within 15 minutes, In the process of germination, the spores absorb water and swell, the protective coat disintegrates, and a single vegetative cell emerges.